Welcome to this Multi-omics data analysis webinar. My name is Jan Nilsson. I'm your host here for this webinar today. And we're going to talk about multi-omics in Qlucore Omics Explorer. Multi-omics is getting more and more popular, and you can do multi-omics using a number of different types of data: transcriptomics, proteomics, epigenetic data, and metabolomic data. Multi-omics can mean quite a lot of different things. We are going to look at correlation analysis with two different types of data in this webinar, but there could be other forms of multi-omics using variable list, variable annotations with protein-gene relations to work with both transcriptomics and proteomics datasets in parallel. Combine a genomic view and an expression view of an experiment, combine ChIP-seq, ATAC-seq data with RNA sequence data, etc. In this webinar, we're going to work with transcriptomics and proteomics data from the same patients to demonstrate how you can identify correlations between genes and proteins. We're going to start by making a correlation matrix using those two data sets here. And we'll assume that the datasets are normally distributed, and we will merge the two datasets in Excel or a similar tool before that, and then we include also a column with information of the type. For instance, a gene, protein type to indicate which dataset it comes from. We'll also look at other types of correlation in this webinar. We'll be looking at regression analysis, we'll be looking at using the Qlucore Biomarker Workbench, so there will be many different examples here, but we'll start with an easy one, a correlation matrix. If the data is organized, like you can see on the presentation to the right there, we have sample IDs and treatment on top, for instance, so one identifier and also one sample annotations. And then we have the gene or protein information where we have the proteins on top here and then followed by the genes. In many cases, the number of genes that you have are quite many compared to the number of proteins that you are looking at. So typically it could look something like this. And I'm just going to import this now, and we will have a look at it using the Qlucore Omics Explorer window and also do a correlation matrix out of the data. It will be something like that you can see at the very bottom here after the export here, and to the right you see the information that we have imported, organized here with a dendrogram to the left. So let's see if we can do this and get it imported using Qlucore Omics Explorer. I select File here, Open with Wizard here, and as before I mentioned that we have our protein data and gene data in the same file for this example here. Press Next here in the wizard. This other type of data here. Press Next. Separator is Tab. Data is organized as a matrix here. Variables here are rows. Data matrix starts here. Then I use the sliders to go to the last sample and the last variable here. Press Next here. Variable annotations, here I got the gene or protein here, and the type, you can see that's protein on the top here, followed by genes further down. And then press Next and bring in the sample annotations also, like this. Press Finish, and we'll see the data set. We can change this into a heat map also, make it a bit bigger for you. You can see it here. And we can organize it also in the way that we order, for instance, the data with hierarchical clustering like this. And we can have auto calculation. And we could also, depending on what type of other information we have, order by some annotation like treatment that we have in this case. So you can see the data here. You can also add colors and so on. Now, if I would like to make a correlation matrix, I can simply go to File, and then I select export, and I select a bit further on variable covariances correlations. I give it a name and call it core. It will be a text file. You select the annotation that you would like to have. We will use the gene or protein names and the correlation here, and then you just press OK. And then a correlation matrix, as I showed in the presentation, will be exported to disk. But there are other ways at doing correlation analysis also. I will go back to the presentation and we'll have a look at that also. Note also that you can import the exported file that we just created here using the wizard if you would like to make a heat map out of the correlation values. Then it will look something like this for the data that we have just exported. Note that you can't do any analysis on it, but you can visualize it, and you can of course export the heat map plot to disk with the correlation values also. Now we'll talk about linear regression, because with linear regression calculation we can add significance also to the correlation values that we have created. And to get those significance values, then we can use linear regression with a numerical sample annotation like time, dose, etc. And in this case here, we have, of course, the proteins that are numerical values. They are variables from the beginning, but we will use them here as sample annotations. We will start by importing the larger dataset only, typically the transcriptomics dataset using the wizard, and then we use variables from the smaller dataset, here the protein data, as sample annotation, and then use linear regression to find each protein statistically significant correlating to genes. So data preparation, we will first import using the ordinary wizard, and then after that we will add the protein values using import sample annotations via wizard. So it's under file import and then sample annotations via wizard. It's, as before, important to have the same sample key, in this case the sample identifier, in both datasets, so we got matches there, so the same protein values for sample 1 corresponds to the gene values also for sample 1. We will then also color the information that we have in a heat map and look at one of the proteins, selected one here, ACE2, and look for significantly correlated genes. And in this case here, we will then also use the information that we have calculated to color the heat map. To the right, you can see the individual values here for ACE2, which is then actually numerical values in the protein dataset, but will be used as a numerical annotation in the transcriptomics dataset. So let's see if we can get started with that, and then we will also set up the statistical test here. We'll set the Q value of 0.01. We will have a look in the variables tab for the Q values and the R statistics values. The R statistics values, that's the same as the correlation values. So it's those values that we are really interested in. Let's see if we can do an import now. I will close this dataset here. And now we will simply import using the wizard the transcriptomics data only. Wizard starts. It's other data. We have a normalized transcriptomics dataset organized as a matrix. I treat rows as variables here. The data matrix starts here. Go to the last sample and the last variable here. Press next. Variable annotations, that's just the gene symbols that we have here. and then the sample information here. Press finish. Now we have only the genes in this data set here, and we will change the things to a heat map also. Make it a bit bigger here, and in the view tab here we will do auto calculation, so we'll see everything. Now I will also import using another wizard. Under file here we have got import, and then we have variable annotations and sample annotations by a wizard, and here it's sample annotations that we're going to import. I select now the protein dataset here. Start that wizard. Separator has been selected there. The annotation information is organized here in columns here for the samples. Press next here. Identifier to use here for the cell that contains the sample identifier is this one on top, and that must correspond then to, of course, to the transcriptomics dataset. Rows. Then I click on the first one here. Then I use Shift for the last one, so I bring in all those proteins here. I'm using the Shift key here to bring both the sample identifier as the identifier and then the rows with annotation here. Click on the first one and then on the last one using Shift. I press Finish. And now the annotations have been imported. Let's go here to Samples, and we've got sample identifier, but you can see that we have got all the proteins as well. We've got treatment also, but all the other ones as well. And we're going to have a look at the first one here as an example here. And you can see the values here are numerical. And we can start also by coloring it using a color range here. So the first one, let's say that we make that into light yellow. And the last one, we make that into red as an example. And then we can use this color as a range here. Color all values based on names or as a gradient. Select OK. Press on the first one here on top, this one. Press OK, and then we'll take the last one here. Press OK, and now we can see that we have colors as a range here from yellow to red. If I would like to put that on top here, we can of course do that as well. So here we can then have the colors here for this annotation, which was called ACE2 on top. And we can order it then based on the names that we have or the values that we have here. So order here for, in this case, the samples here could be according to ACE2 and numerically. Then we can, of course, also add information on the right-hand side, but first we'll set up a statistical test. As I said, we can run a statistical test here using one of the proteins as the annotation. So we'll do a regression test here. Select it here as linear regression because it's, as you can see, numerical values. Using then the ACE2 protein here. And we can set here a Q value, a threshold value. Let's say that we set 0.05 or something like that. Here on the variable side here, we can also color according to the R statistic value here. And we can then order according to that also. So we have the ordering here according to the value that has been calculated, in this case the statistic value. And if you would like to see the results, we can go to the Variables tab here. We have got all the different variables that are the genes that we're looking at here. And we have now done a correlation between the protein ACE2 and the genes here. Let's see, we can present here if we would like the R statistic value, which is then the correlation value. You can mark it from the highest positive one and then to the highest negative one at the bottom. We can add also a Q value so that you have that as well. So then for each of the correlation values, we'll see the significance value, also the Q value that we have here for, in this case, the correlation in between the ACE2 protein here and the FADS3 gene. And of course, all that information can now be exported using this icon here with a tray and an arrow going out from the tray. This is how you can do it for one of the proteins here, but we can also take advantage of using the biomarker workbench for this, that can work with multiple proteins and correlation to the genes. So let's go back to the presentation and have a look at that also. One thing that I can mention also is that if you would like to have the information presented in another way, you can set up a scatter plot, a scatter sample plot here, and then you can, for instance, select the ACE2 protein here and those genes that correlates best to them, both from a positive correlation and a negative correlation. And this is how you can set it up. On the x-axis, you select ACE2 numerical value, and then on Y, you simply click on those genes that you're interested in, those with the best correlation value. And then the values are presented in a scatter plot like this. The Biomarker Workbench is a tool within Qlucore Explorer that can be used for multiple tests. It's very powerful. It can be used for many different purposes. And here we are going to use it to get the correlation values for a number of proteins. As you remember, we imported several proteins. And the calculated R statistics value that we'll get here will then be for those proteins that we have correlated to the genes. And we'll also get the P and Q values for the significance of the statistical test. You launch the Biomarker Workbench, and the launch, this symbol here is the Biomarker Workbench, and it will then start up, and you will see how it looks like on the left-hand side here. There are a number of icons where you can specify which test that you would like to run. They look like this, a plus sign if you would like to set up one. If you would like to set up several and get a proposal, then you use this icon, and we'll see how that works. Normally, for efficiently, you import a maximum of 100 annotations proteins in one batch in order to use the Biomarker Workbench. So if you have several, then you do several batches when you do the import. So this makes it more efficient running on a normal computer, like, for instance, mine, a laptop here. And here we will see how we can set it up using this proposal icon, and then just press calculate to get results. In the result part, you will get both an overview and a detailed view, and we'll have a look into the detailed view where you get the R statistics value, and we can select which columns that we're interested in. There is a column selector here in the details view, and you can select among those things here like P and Q values or statistic value that we're interested in here, for instance. There is also an export, so you can export the information out on disk and import it, for instance, in Excel if you would like that to order things in. And here is a selection then, and as you can see here, you can press export and a full export. Export is those things that you see. Export is also the other columns that you haven't specified, but it exports all of them. I will now leave this, and we will see how we can work with the Biomarker Workbench. So back into the program again here, bring back all the information here. And now I will simply start the Biomarker Workbench here. I will use the same information, so we have our samples here. That's all of the proteins here. They are less than 100 here. And then as the variables here, we will have our genes. Those could of course be many, many more. Let's see now if we can start in the biomarker workbench instead of using a single test here. The biomarker workbench looks like this here. And here, as we said, we can either set up a single test, just like we did in the statistics window, but there is also an option here to set up many. I will make this window a bit bigger for you, so it's easier for you to follow also. Let's see now. I select this icon here, which has got a plus sign, a number of sheets. Click on that, and it will then propose tests. And since it's the numerical values that we have for all the proteins here, you can see that it proposes linear regression for those, except treatment, which is more of an ordinary sample annotation. But the other ones are linear regression tests. The test has not been run yet, but when I press calculate here, it will start to run the test. And you can see the values calculated here, the number of variables that satisfy the threshold criteria here. If I now go to the details view, all the tests here, we can see that we have here response variables that we are looking at here, and that's the genes that we have in the transcriptomics data set. Here, those columns here represent, as you can see, ACE2, the protein that we worked with before, P and Q values, and the next protein, etc. I will change what we see here in the columns. So I will add the R statistic value, and I will remove the P value, so we just have the Q value, and I think I will also remove minimum values. So what we see now is that we have here the R statistic value, which is the correlation value for the ACE2 protein. If I click here on the column head, you see the highest positive value here, and that seems to correlate with FADS3, that gene. So the best correlation is this one, and you can see that you get also a good Q value. If I go to the next protein, this one here, I can click here on top here, and then I get the highest positive correlation value here. You can of course go for the lowest one as well, and we can see that the best correlation is with this gene here, and so forth. So all that information is available in the columns that we have here for each of the proteins in the calculated matrix here. If I would like to export all of this, you just press export if you just want those columns, or a full export if you would like to have the columns that are not visible at the moment. But if it's just the ones that you would like to see now, then you press export, and then you have all that information. And if you would like to, you can just go into the details in, for instance, Excel. It will be exported as a text-oriented file. Let's go back now to you can also, if you would like to take a single test here, like a test like this, and then you can copy it to statistics, so you can do an individual test from the biomarker workbench into the ordinary program as well. Copy to statistics in those cases. Let's go back to the presentation again. So now we have seen how we can use the biomarker workbench for easy multi-omics, where we do correlation analysis on many proteins in this case towards the transcriptomics data that we have. It's two separated data sets where we use the transcriptomics data, which is likely to be the bigger one, as the data set, and we're using the proteomics data set as sample annotations to do linear regression in between a protein and all the genes in the data set. And we can work in the biomarker workbench in batches of around 100 proteins. That's all that I'm going to cover today. If you would like to learn more, you can contact us at Qlucore, and you can of course also read more about this at our